As I mentioned, proteins work in three dimensions rather than sort of sequence strings like we're used to. But sequence strings are useful because they're easy to compute and they're fast. However, what's important to note is that if sequences have diverged from each other, which we can see from a multiple sequence alignment, it tends to indicate they're probably not that similar to each other. So structures actually can reveal relationships that aren't necessarily evident from primary sequence because of this reason. So if you were to look at primary sequence, and the example I'm going to show you here, actually, I'll just show you, is between two proteins. One is called MREB in bacteria. The other, hopefully you're aware of, it's a cytoskeletal filament known as actin in eukaryotes. And actually, obviously within muscle contraction as well. What you'll note, well, what I'll tell you is, they don't share very much sequence identity. So in this case here, less than 15% at the primary sequence level. We can't really detect this, therefore, and they wouldn't come back as, you wouldn't think they were homologs at all. But actually, when we solved their 3D structure, and you can see they're binding to ATP here in yellow, they're very, very, very similar. And actually, they are therefore relatives of each other, but it wasn't clear when looking at primary sequence alone. We had to know about structure. And as soon as these structures were solved, their evolutionary relationship therefore became fairly obvious. But actually, being able to predict protein structure is not a trivial matter at all. And really, there are caveats to this slightly, but it's still a very, very tricky process. And actually, I would say we're still unable to do it accurately enough. And that is because there are so many different structures that the same sequence will potentially be able to fold into. So, if you take a standard protein, for example, actually, there's lots of possible conformations. And for a typical protein, it's probably, 10 to the 140, something like that, 143 from memory. So that's, one with 143 zeros behind it. That's how many different possible conformations you can have. So this is a kind of example. If we were to take this amino acid sequence here, right, there's only 6 amino acids, but actually you can see they're present within both of these proteins in purple. One is within an alpha helix, the other is within a beta sheet. So you can see that actually secondary structure preference is really not fixed, and therefore predicting just from a sequence like this what it's going to fold up into is really difficult. Protein folding is incredibly complicated. So we are unable to predict using first principles what sequence will kind of fold into. And there are various other chaperone proteins and various other things that feed into that. So therefore, how do we kind of predict structures? Well, first of all, we need to rely on solved structures. So structures, something like this, where it's a solved structure, is done so via X-rays and X-ray crystallography. It's really, really hard to get this data. Actually, we're still trying to do it in the lab for one protein. It's really hard. Some proteins don't want to crystallize. It's just problem after problem. Even if you do, actually analysing that data and getting it correct is really tricky as well. As it says here, it takes years to solve a crystal structure. And obviously not all types of protein are amenable to these methods. So how do we infer structure? Well, we do it using something known as homology modelling, which is to take a probable structure based on, make an inference of a probable structure based on one that we already know. So we could do a multiple sequence alignment like this, and it would be the case that we could take something like the worm structure that perhaps we have a solved structure for using X-ray crystallography, and it's all good. We could then try and fit the human sequence to that model, and kind of make a sort of guess as to what it would look like if it folded in the right way. But that's important. So this does not predict, right? Homology modelling does not predict structure. It addresses the question, what would our protein look like if it folded similarly to, for example, here, the worm protein? what would it look like if that were the case? Okay, so it's not a prediction of what the structure is. It addresses this question down here. But the important point is that any solved structure we're kind of relating our sequence to has to be related, sort of evolutionarily. It has to be ideally, very similar, but it also has to be solved, and it has to have been solved by something like crystallography. So where do we get this information from? Well, if we go to a database known as the NCBI, which I talked about, but this time you can click structure on this menu rather than nucleotide or protein, and you can type in a protein, the name of a protein. You can search using the same fields and Boolean operators, and then you can gain all these structures here, and you can download various files to then be able to view them later on. You can also use the protein data bank, which is what we're going to use. Again, you can find a particular protein. They all have their own little codes here. And actually you can download the files by clicking this arrow, and you can download the relevant file to then put it into something called PyMol, which will allow you to interact with this image and highlight different things. This is the PyMol viewer, and you'll become more aware of this during the practical, so I'm not going to focus too much on it. But we've got an external GUI, or graphical user interface here. Don't worry too much about that for now. Your menu bars up at the top here with various functions. We have a command line prompt, so you're going to be using that to type some stuff in. The main viewer is here, so the protein will appear in here in 3D, which is the display area. You have your control panel here with these A, S, H, L, and C tabs. That is explained within the instructions that I've written for you, so don't worry about that for now, but you use these to colour different parts or select different parts or hide waters or show waters or turn your image into like a stick model, ball and stick model rather than a ribbon diagram, etc. So we've got various control buttons here. So actions is A, show, hide, label, colour. That's what these stand for. We're not going to actually worry about this, but this actually tells you, and you can actually change what controls your mouse uses, and you can make movies as well. So you can actually spin this round and make a film, which takes a while. So we're not going to do it, but you can, if you wish, look into that. So movie controls there. There's another line command prompt as well, which again we'll be using during the practical. So next time, it's not necessarily next week, but it's whenever we meet next, is the final computer practical. Please make sure you download PyMol. Again, the link is in the handbook and on Teams, but this is the website. It is free, so just download it for the next practical. And the same thing applies. Okay, so we'll open the last computer lab. The instructions are in there with the quiz, and I'll be on hand to ask or answer any questions.